Hi, welcome back to Part B of Chapter 1 as we talk about the rest of the chapter. Here we'll talk about the two broad categories of cells, prokaryotes and eukaryotes, and then we'll talk about model organisms and some important things about how we study cells. So prokaryotes are the most diverse and the most numerous cell type on Earth. Prokaryotic cells have some characteristics. As you know, they have an outer membrane, but they don't have any internal membranes, no compartmentalization by membrane structure. They have a much smaller genome than eukaryotes. The genome is contained in a single circular chromosome. So among the prokaryotes, we would categorize these cells as either bacteria or archaea. There are some important differences between the two. Archaea were first discovered in very extreme habitats, and so even though each type of archaea has a particular habitat in which it lives, we can find archaea in general in very extreme places as well as in some very common places. There are archaea that live in your gut, live on your skin, as well as at the bottom of thermal vents and in places where there's no oxygen and where there's very high salts and so on. Bacteria are found in more common locations, but they're spread according to each different type of bacteria's properties. They're spread in different places as well. So you find them in great diversity of places. The difference between an archaeum and a bacterium would be the composition, the chemical composition of their cell wall, their cell membranes. Also, they have specific enzymes that are unique to archaea versus bacteria, and archaea have some unique metabolic pathways that are not like archaea. Both archaea and bacteria exist primarily in these three different shapes, although they're not the only shapes that you would find. Some are spherical, like the strep. Some are rod-shaped, like E. coli and Salmonella. Others are spiral, like the Treponema that we see here. One bacterium that everybody needs to know about would be E. coli. This is a common organism, a common bacterium that lives in the gut, lives in feces, but it also has served as an important model organism. We've been using E. coli to study important cellular functions for a long time, and very much is known about them. When we talk about eukaryotic cells, now we're talking about cells with a larger genome contained in numerous chromosomes that are linear and a bunch of membrane-bound compartments inside. These compartments we mostly call organelles. So the nucleus is where information is stored. Mitochondria generate usable energy. Chloroplasts capture energy from sunlight, and so on. These are categories in your textbook. There's quite a bit of information, but the truth is, as we go through the entire semester, we'll be hitting on all this information in great detail. Right now, I'd just like for you to have some basic foundation of what each of these structures is like. Here we have a line drawing as well as an electron micrograph of the nucleus. You can see its double membrane. Here, of course, is where most of the DNA in a eukaryotic cell is contained. Mitochondria are also double membraned, but the inner membrane is very highly infolded to give it a tremendous surface area. You probably know mitochondria as the powerhouse of the cell, and those important protein complexes, enzymes of various types that are involved in using oxygen in order to be able to make ATP from nutrient molecules, those are housed in the mitochondria. On those inner membranes are some of the most crucial ones for the final steps in producing ATP. You've probably all heard of endosymbiont theory that describes how the similarities of mitochondria to aerobic bacteria are used to explain how mitochondria may have come to be. So the idea is that early anaerobic eukaryotic cells were probably predatorial, took up an aerobic bacterium. The capability of this aerobe to produce a lot of ATP quickly was, of course, beneficial to the host cell, and ultimately that bacterium became a mitochondria with a permanent home inside the cell. A mitochondria, of course, can reproduce independently of the host cell. They have their own genome, and they have their machinery for using that genome to make proteins. The next organelle we'll look at are chloroplasts. Chloroplasts also have not only an outer membrane, but several types of internal membranes. They contain chlorophyll, a pigment that absorbs light energy, and use that energy of sunlight to create nutrient molecules. Chloroplasts are similarly thought to have arisen from the predatorial ingestion of a photosynthetic bacterium this time. So the bacterium, rather than being an aerobe, was a photosynthetic bacterium and developed into a chloroplast. So eukaryotic cells have numerous other internal membranes, so they're highly compartmentalized inside. The compartments have a variety of different functions. Here we can see in this electron micrograph a cell that has very extensive endoplasmic reticulum. Cells that secrete proteins oftentimes have this kind of an appearance. You notice the numerous ribosomes that stud the membranes of the endoplasmic reticulum. So the proteins that are first assembled as sequences of amino acids are processed within the endoplasmic reticulum, moving them toward their final functional state. Another internal membrane structure would be the Golgi apparatus. It looks like a stack of flattened discs here. The partially processed proteins are passed on from the ER to the Golgi apparatus, where further processing is accomplished. And from the Golgi apparatus, these proteins are distributed to their ultimate location, whether it's inside the cell, whether it's to become a part of the plasma membrane, or to be secreted to the extracellular space. These membrane-enclosed organelles are distributed specifically and held in place in locations where they need to be within the cytoplasm of the cell. Cells are constantly in communication with their internal and external environment, so one of the ways in which they're able to communicate and influence their environment is by importing and exporting materials using membranes to mediate the traffic. There are other mechanisms as well, but the membranes are an important component of this important trafficking. The background material in which these membrane-bound organelles are embedded is called the cytosol. The cytosol is an aqueous gel. It's extremely crowded with large and small molecules as well as ions in the watery matrix. Another component of a cell that's important to know about would be the cytoskeleton. These consist of different types of structural proteins that are organized in specific ways and attached to membranes or other components of the cell in order to give it structure, to form things like microvilli, and to direct cell movements, whether it's the whole cell migrating from one place to another, or whether it's the movement of materials within the cell, or adhering an organelle in a specific location. The three categories of cytoskeletal proteins are seen here. Using fluorescence spectroscopy, we can see the actin part of the cytoskeleton, the microtubular part of the cytoskeleton, and then the third category, intermediate filaments, depending on the cell type. These may actually be different kinds of proteins. Here we can see mitotic spindle, and the process of mitosis depends upon the interaction of microtubules with chromosomes and the stabilizing force of the actin filaments that are associated as well. Now we'll move on to discussion of model organisms. As we said earlier, molecular biologists have focused primarily on E. coli, such as we see in the upper left-hand corner. This is the same image you saw a few minutes ago. Brewer's yeast is a common model organism. It's useful because it's a simple eukaryote. It has the properties of a eukaryote, but doesn't have some of the complexities of other types of eukaryotes. Arabidopsis in the bottom is a plant species that has been chosen as a model plant. Model organisms, there are many model organisms. They include flies like the Drosophila in the lower left, the C. elegans that you see to the middle right, and the zebrafish that you see in the lower right. This is not an exclusive list of model animals. Mice are another. When you take human cells from the organism and grow them in culture dishes, if you give them the right environment and the right nutrients, they can exhibit some of the properties that they would also exhibit were they in vivo, were they in the organism as such. So fibroblast cells, for example, characteristically adhere to their substrate, and you can see the shape of the fibroblasts on the left as they have these extensions that stick to the plastic of the plate. Neurons are known for their spontaneous ability to interconnect with one another. Of course, neurons are communication cells that rely upon interconnections, and you can see them developing in culture. And in part C, we have epithelial cells, which form sheets. Epithelial cells are known as covering and lining cells, and this sheet formation is characteristic. We can also learn about the genomes of different types of organisms. We can compare whole genomes, and we can compare individual genes that code for specific proteins. By doing this, we can reveal specific differences among different types of living things, and we can also identify commonalities, even things that are common to everything that's alive. And we have our whole semester ahead of us to expand our understanding of all these things that we've just briefly mentioned as we've gone through this overview first chapter. See you soon.